All right, in this set of slides, we're going to continue to talk about different aspects of carbohydrates, and here we're going to discuss polysaccharides. So we're going to discuss a number of different polysaccharides, including cellulose, chitin, and starch, as well as discussing a particular class of them called glycosaminoglycans. Before this, we talked about carbohydrate functions, as well as monosaccharides and disaccharides, and in the next set of slides, we will talk more about glycoproteins. But for now, let's go on to talk about polysaccharides. So two very important structural polysaccharides that we're going to talk about now are called cellulose and chitin. So they're very similar to each other, but they do have some important differences. So first of all, their functions are a little bit different where we find them. So cellulose is a component of the plant cell wall, whereas chitin is part of the exoskeleton of invertebrates. They are both made of units of a single monomer that's repeated over and over. So for cellulose, that single monomer that we see repeated is glucose, D-glucose specifically, whereas in chitin we have N-acetylglucosamine. So if we recall from the last set of slides, this N-acetylglucosamine is actually the same basic sugar as glucose, except the difference is there is an acetyl group attached to an amine, and it's on the second carbon. And usually when you see an acetyl or an acetylglucosamine like this, even if they don't specify that it's on the second carbon, it's usually you can assume that it's carbon 2. You can also see that the type of glycosidic bond is the same in both of these sugars. So let's think about how we can name this glycosidic bond. So let's start at the top with cellulose. So let's start numbering the carbons in our glucose sugar. So carbon number 1, that's our anomeric carbon. We have 2, 3, 4, 5, and the 6th one pointing up. In our second sugar participating, which is also glucose, we have the anomeric carbon over here participating in a different glycosidic bond. And then we have the 4th carbon that is participating in this glycosidic bond. So how do we name this? First, we have to consider the position of the hydroxyl group on the anomeric carbon in our glucose on the left. And this hydroxyl group is pointing up. Recall that these bonds are drawn as curved just for ease of drawing to make it all fit in one spot, but remember in real life this bond is straight. So if we were to draw out this bond, it would really realistically look more like this, with this part of it going to the second glucose. At any rate, we know that this original hydroxyl is pointing up, so it's beta. We know that it's on the 1 carbon, the anomeric carbon, so we would call this beta 1. And then the second number we would use to describe this glycosidic bond is where that bond is occurring on our second glucose molecule that's shown here on the right. And we see that it's attached at carbon number 4. So we can call this glycosidic bond a beta 1,4 glycosidic bond. And this particular beta 1,4 glycosidic bond is not digestible by vertebrates. There are other organisms that can digest these types of bonds, and they use an enzyme called cellulase. But vertebrates, like us, we lack those, so we cannot ourselves personally digest cellulose. If we look at chitin on the bottom, let's think about what type of glycosidic bond we have here. So as we mentioned before, this is the same exact sugar. It's based on glucose. Its numbering is going to be the same. And let's just actually take a moment here and make sure we have the same sugar. Don't just take my word for it. And to do that, we can look at the configuration of each of the carbons in our ring. So because they're drawn in the standard sort of orientation, you know that if you see all the groups pointing in the same direction, you must have the same ultimate configuration. So for glucose, we have our anomeric carbon. It's either alpha or beta. In this one, it's beta. In our glucose on the bottom at our anomeric carbon, we also have our hydroxyl group pointing up. It's also beta. Our carbon number 2, hydroxyl, is pointing down. Same thing in our chitin molecule. In this case, the hydroxyl group has been substituted with an amine, but you get the idea, it's pointing down. In our 3rd carbon, both these molecules, the hydroxyl group is pointing up. 4th carbon, it is pointing down. It happens to be participating in a glycosidic bond, so it looks curved, but you know it's pointing down. And then in our fifth one, it's going to our sixth carbon, which is pointing up. So ultimately, now we know these are the same exact sugar, right? The one difference is that substitution on the 2 carbon with the N-acetyl group, with the N-acetyl, that's what we see in chitin. And so again, in chitin, just like in cellulose, we have that beta 1,4 glycosidic bond, where if we consider the sugar on the left, we have our beta hydroxyl on the 1 carbon sticking to the 4 carbon on our adjacent sugar. All right, so let's keep talking about cellulose and chitin. So like we mentioned, they have all of these components and they're very structural, right? So we know that they're going to form these long linear chains that we saw in the previous slide linked by glycosidic bonds. We also can see that they can sort of form sheets in a way because you can have hydrogen bonding between, we have all of those hydroxyl group on all of those sugars, as well as the nitrogen as part of in the case of chitin. And so as we recall from a few chapters ago, if we have everything lined up properly, you can form hydrogen bonds, which are pretty strong and can help these linear sort of change of sugars to become more sheet-like, or just more. They can form structures that are extremely strong. All right, so now let's talk about something similar but different. So let's talk about starch. So starch is also a polysaccharide, but starch we think of as more of an energy storage. If we just to go back to cellulose and chitin, they are structural, right? When we think about starch, it's really for energy storage because we can break down the components of the starch into monosaccharides that we can ultimately break down and use to help us make ATP that we can use for energy purposes within our cells. So let's think about starch. So starch is something that is produced by plants, and again, it's used as energy storage. So one type of starch that we're going to start talking about is called alpha amylose, and we see that pictured here on the right. Alpha amylose is completely linear. It is just a repeating chain of glucose over and over and over. So just one type of sugar molecule, glucose, it's repeated, and the one thing that might look different here than some of the other disaccharides and polysaccharides that we've looked at before are the type of glycosidic bonds. So let's figure out what type of glycosidic bond we have here in alpha amylose. So first we'll start again, number our carbons. So we have carbon number 1 here, that's our anomeric carbon next to that O that's in the ring. We have carbon 2, 3, 4, 5, and 6. Same thing with our second sugar. So now if we start with our first sugar, we have to first consider if we have an alpha or beta position on the anomeric carbon. And so if we look at our hydroxyl group, it's pointing down. That means we have an alpha position in this case. And again, it's on that anomeric carbon is the 1 carbon. And where is that glycosidic bond going to? Again, it's going to the 4 carbon on the next glucose. So now we have an alpha 1,4 glycosidic bond. So yeah, that's alpha amylose. It's entirely linear, it's entirely made of glucose, it entirely uses alpha 1,4 glycosidic bonds or glycosidic linkages to piece together those glucose molecules. And so alpha amylose is similar but different to amylopectin. So amylopectin essentially starts as alpha amylose, but it has the addition of these branches that you see pictured here. So approximately every 25 glucose molecules, there is a new branch. So like we mentioned, the linear part of it that you would see extended along left and right, it looks just like alpha amylose. So this would consist of alpha 1,4 glycosidic bonds. But we also have some branches that we see pictured on the top here. So what type of glycosidic bond is in this branch? It's not alpha 1,4, it's a little bit different. So again, let's take a look at our glucose. We have our glucose in the alpha configuration because it's pointing down, and that's on that anomeric carbon, carbon number 1. And just for the sake of completeness, I'll finish numbering these carbons on our first sugar with that anomeric carbon participating in the glycosidic bond. Our second sugar We can also number those carbons, so anomeric carbon, number 1, making our way around the sugar. And now we can see that this glycosidic bond from this first carbon is being linked to carbon number 6 in our second sugar. So that makes this an alpha 1,6 glycosidic bond. So yeah, so when we look at what does this mean when we look at the larger structure of each of these molecules? So again, as a reminder, alpha amylose is linear and it's all glucose, and it only has that one type of alpha 1,4 glycosidic bond. And ultimately, this means that alpha amylose a lot of the times takes on this sort of coil shape because it's linear and because this is a relatively stable configuration for it to end up adopting, or conformation, I should say. And this is a little bit different when we compare it to amylopectin. So recall that amylopectin has those branches that are derived from those alpha 1,6 glycosidic bonds. Those branches occur approximately every 25 glucoses. And so what you'll end up seeing looks a little bit more like, like something with branches, like a tree or something that you can imagine like that, where you have all of we have all of these linear bits, but there are all these branches coming off of them. And it's relatively spread out. It's not able to be as sort of compact and coiled as alpha amylose might be. So what is going on with starch? So we talked about before how cellulose and chitin are structural. We cannot digest them. But we do have the capability of digesting some starches. So how do we digest starch? We can use different enzymes that can break those different glycosidic bonds for us. And doing that, as we recall, starches are made of individual glucose molecules. If we can free those glucose molecules, we can, again, use them in other processes that will ultimately help us get energy out of those glucose molecules that we can use for other cellular processes. So what are some of those enzymes that help break down starches? One of them is called amylase. Amylase is found in saliva as well as the small intestine, and it hydrolyzes some alpha 1,4 bonds. And again, we see those both in alpha amylase as well as amylopectin. And so once amylase has worked on some starch that you've eaten, it hydrolyzes some but not all of the bonds, and so you end up with small oligosaccharides, which are relatively short chains of sugars, unlike these long polysaccharides that you would consider starches. So how do we start to cut apart those oligosaccharides? We have another enzyme called alpha glucosidase. So this enzyme hydrolyzes those remaining alpha 1,4 bonds. Again, same bonds, just now it's turning those oligosaccharides into glucose monomers. But in the case of amylopectin, that's not enough, right? We have those other alpha 1,6 bonds that create those branches. And so for amylopectin, we need this debranching enzyme. So debranching enzyme is able to hydrolyze both alpha 1,4 and alpha 1,6 bonds. And so it's going to help us make glucose monomers out of amylopectin. So amylopectin and alpha amylase are found in plants, like we talked about. What do mammals do? What do animals do? So animals like us have a different form of energy storage, polysaccharide, and ours is called glycogen. In humans, we find it mainly in skeletal muscle as well as in the liver. And it actually has a very similar structure to amylopectin. It is also entirely made of glucose. It primarily has these alpha 1,4 glycosidic bonds, which helps it start out linear, the way alpha amylose is. But glycogen also does have some alpha 1,6 linkages that result in branches. And glycogen differs from amylopectin because of how frequently you see one of those branches. So for amylopectin, we saw them approximately every 25 glucoses. In glycogen, it's more frequent, so we see them about every 10 glucoses, and so that results in a difference in the way they appear. So in amylopectin, you see these longer linear bits with relatively less branching. Glycogen is much more branched and spread out. And because of all of this more frequent branching, you have a lot of glucoses that are located on the end of a branch, which makes them more readily available to be released when you need a single glucose to be used, when you need more energy and you're in a hurry. So how do we cut those glucoses off of glycogen? So in animals, we have a different set of enzymes that specifically breaks down glucose. So one important enzyme is called glycogen phosphorylase. This is going to hydrolyze those alpha-1,4 bonds that are part of those linear chains. And we also need a debranching enzyme because we also have alpha-1,6 bonds. So our glycogen debranching enzyme is going to hydrolyze those alpha-1,6 bonds that are found at the beginning of every branch point. And again, like I mentioned, the larger number of ends that we see because of that frequent branching allows for fast access to glucose when we need it. So now let's switch gears a little bit and we'll talk about another class of polysaccharides called glycosaminoglycans, or GAGs. GAGs are frequently found in extracellular matrices. So what are GAGs? They are unbranched polysaccharides, so they're entirely linear. And they are composed of repeating disaccharide units. So the first sugar in that disaccharide is always a uronic acid or a galactose. So if we recall, a uronic acid is the naming convention for when we oxidize that final alcohol that's on, as we see it on a linear sugar. And so that's usually carbon number 6. So in our example here, we have carbon number 6. They used to be an alcohol here. Now it has been oxidized to a carboxylic acid. In the second sugar in that repeating disaccharide, it is always a hexosamine. So hexosamine meaning a sugar that originally had 6 carbons, and it's an amine. It has an amine substitution, and even when it's not mentioned by number, it's almost always on carbon number 2, just like we see here. And in fact, the 2 hexosamines that we specifically will see on GAGs are almost always glucosamine or galactosamine. So if we see here, these GLC, that means glucose. GAL, that means galactose. And NAC means N-acetyl, whichever sugar amine. So N-acetyl glucosamine or N-acetyl galactosamine. So what are some properties of GAGs? So because they have all of these charged and polar groups all over them, they really, really like to interact with water. So this can make them really viscous because they really like to take on water and get nice and gloopy. They also have this property of elasticity, and so one way you can think about that is they really, really like interacting with water, and so this makes them sort of compressible, because if you can compress them and maybe remove some of the water. But at a certain point, because of all of these similar charges, all this sort of negative charge character, when they get too close, they'll start to repel each other, and that makes them sort of bounce back to their original size or shape. And so that's what we think of when we refer to the elasticity and viscosity of glycosaminoglycans. And so ultimately, this helps GAGs form these highly hydrated gel-like matrices that are really effective for protecting and interacting with the outsides of cells. So what are some specific examples of GAGs? So first example we're going to talk about is hyaluronate. So the 2 components of the disaccharide repeating subunit that we see in hyaluronate are glucuronate and N-acetylglucosamine. So again, glucuronate started out as a glucose, but we have this oxidation to make the uronic acid. So this would be glucuronic acid or glucuronate because it's charged. And the second sugar we have is N-acetylglucosamine. So it started out as glucose, but we have this substitution on carbon number 2 of an amino group that further has been acetylated. So we have an N-acetyl D-glucosamine, which again we can also write as GLC NAC, N-acetylglucosamine. So when we talk about hyaluronate, it is composed of anywhere from 250 to 25,000 disaccharides. So these are these extremely long chains. And so this results in really highly hydrated molecules that form very viscous solutions. Again, because of those properties of all of that charge that you can try to squish together, but it'll bounce back. It really likes to interact with water, all that good stuff. And so this helps to make hyaluronate a really effective shock absorber and a lubricant. And we find it in our bodies in connective tissue, in synovial fluid, and in the vitreous humors of the eye. There are a few gags that are the major components of cartilage. So these gags that we find in cartilage usually are composed of 50 to 1,000 disaccharides, so a little bit shorter than hyaluronate. The first one that we'll consider is called chondroitin sulfate. So the two components of the disaccharide subunit that makes up chondroitin sulfate, the first one again is the glucuronate. So we start out with glucose. We have that oxidation of carbon-6 to the uronic acid, making our glucuronate. On the right, we have an acetyl galactosamine. So before we had glucosamine, now we have galactosamine, which again is abbreviated like this, and acetylgalactosamine. And this sugar has been further modified by the addition of a sulfate group at the 4 position or the 6 position. So at the top we have our N-acetylgalactosamine 4 sulfate, where you have a sulfate on the 4 carbon. You can also have that sulfate group located on the 6 carbon, as is pictured below. And so either one of these disaccharides are considered chondroitin sulfate. So we can have chondroitin 4 sulfate, where the N-acetylgalactosamine is substituted on the 4 carbon, or chondroitin 6 sulfate, where the N-acetylgalactosamine is substituted on the 6 carbon. And the other component of cartilage that we'll talk about now is keratin sulfate. So here we are similar but different. So our first sugar, and all the other ones that we looked at so far, was glucuronate. Here we have just plain galactose. So we do not have an oxidation on that first sugar there. Our second sugar looks a lot like we saw in chondroitin sulfate, where we have, but instead of a galactosamine, like we saw in chondroitin sulfate, in keratin sulfate, our basic sugar here, the original sugar, was glucose. We added that N-acetyl over on the 2 carbon, and we have a sulfate group on the 6 carbon. So altogether, that sugar with all the modifications is now called N-acetyl D-glucosamine 6 sulfate. Because we have our glucose, again, we have on the 2nd carbon, we have the amino, which is further modified with the acetyl, and finally on the 6th carbon, we have that sulfate group, which is, again, highly charged. And so something interesting about keratin sulfate is it actually has a variable sulfate content, meaning that not every single N-acetylglucosamine has a sulfate group. Some of them do, some of them don't. And occasionally you'll also see other sugars in there aside from glucose. So it's a little bit more variable than something like chondroitin sulfate, which has its own variability with the 4 and 6 position, but keratin sulfate has these ones that I just mentioned, with the sulfate content and the incorporation of other sugars. So our next example of the glycosaminoglycan is heparin. So heparin has a very different sugar that we haven't talked about as the first sugar, and that is L-iduronate 2 sulfate as the first sugar. So again, we know it is in the L configuration because the 6th carbon is pointing down in the standard configuration orientation, and we have the uronic acid, or the urinate, because it's charged, because this carbon is oxidized to the carboxylic acid. And at the 2 carbon now, we have a sulfate group. So altogether we have L-iduronate 2 sulfate on the left. Our sugar on the right is one that should look a little bit more familiar, but a little bit different. So our base is glucosamine, right? So we have our amine group on the 2 carbon, but instead of an acetyl group modifying that amino group like we saw before, now we have a sulfate group there. So we have N-sulfo instead of N-acetyl like we saw before. So N-sulfo, D-glucosamine. And then we additionally have a sulfate group on that 6th carbon of that sugar. So altogether for that second sugar, it is called N-sulfo D-glucosamine 6-sulfate. And so altogether, you can see that there are a whole bunch of different positions where you might see sulfate groups. There are 3 different potential sulfates for every disaccharide unit, but heparin is variably sulfated. So on average, each disaccharide pair of sugars has 2.5 sulfates. So some have maybe half have 3, half have 2, maybe a few have 1, that kind of thing. And in addition, we have that charged group on that oxidized 6 carbon on that first sugar. So this sugar is extremely charged, right? We looked at all those negatively charged groups. And in fact, this is the most highly charged polymer that we see in mammalian tissues, which makes it really, really just love water. And it is not found in connective tissue. It actually has a different function that I will talk about in just a moment. But first, I just want to draw your attention to this more 3D model on the bottom here. And this really helps you see all the negative charge that's surrounding this heparin molecule. So it has this sort of helical shape that you can see, and you can see all of these red and yellow groups. Those are the sulfate groups, right? The yellow is sulfur, the red is oxygen, and they're just covering this molecule. It's extremely highly charged. You can also see in blue, peeking through in a few places, that's where the amine is, the nitrogen, and the the carboxylic acid acids are a little bit harder to see in there because they kind of blend in with the background, but again, just looking at this picture can help you really to see how extremely charged this molecule is. So what is its function, right? So it's not found in connective tissue. Actually, what it does is it inhibits blood clotting. So what happens is when tissue is injured, heparin is released from the arterial walls. So generally of course, when we have an injury, we do need some blood clotting to prevent, a massive amount of blood loss. But we don't want too much clotting, right? Because then we could have clots that end up getting loose and traveling through our bloodstream and causing trouble elsewhere. So we have, this really intricate clotting cascade that controls this. And part of controlling this whole clotting process is this molecule heparin. And heparin actually inhibits 2 of the enzymes that are involved in clotting. They are called thrombin and factor 10A. And if you want to know more about this, you can look at box 11.4 in your textbook. It has a lot more information about the clotting cascade for you, which we won't be going into in detail here. So with that, now you are ready to answer the Chapter 8 Polysaccharides practice questions.